Hello and welcome to On the Line. I'm Carl Richter. This week's interview is with virologist and science communicator Dr. Benjamin Neuman, who until recently was chair of the biology department at Texas A&M University, Texarkana. We spoke about the fascinating science behind the COVID-19 pandemic, the virus that caused it, and the vaccines that promise to bring it to an end. First, here's a look at some of the stories we covered this week in the Texarkana Gazette. A Department of Transportation grant of more than $880,000 may make possible twice daily direct flights to Houston from Texarkana Regional Airport. Tentative plans are underway for United Airlines to provide the service, but a contract has not yet been negotiated. The grant funding would be used for a revenue guarantee, marketing, startup cost offsets, and fee waivers. Pier 27, a beloved Doddridge, Arkansas restaurant, has reopened under new ownership. Former owner Jerry Sewell first opened the restaurant in 1984, and it closed in 2015. Pier 27's new owners, John and Angie McAdams, say its decor and original recipes have not changed. The restaurant will be open Friday and Saturday evenings. The Texas Nurses Association asked state residents to do their part in fighting COVID-19, above all by getting vaccinated. The group said nurses are burned out and many are leaving the profession as hospitals near capacity amid a spike in infections driven by the Delta variant of the coronavirus. A clear pattern has emerged of unvaccinated people contracting COVID, and most hospitalized patients are also unvaccinated, according to the association. After a break, this week's interview. Subscribe today to the Texarkana Gazette and you'll get an Apple iPad to use at no charge, plus a Sunday paper delivered to your home. Every day you'll get an electronic version of the Gazette downloaded directly to your iPad. It's the ultimate home delivery. No more waiting, wet papers, or going out into the cold. And everything you want, the latest news, puzzles, and coupons, is right there. You can zoom in to make articles easier to read, or have the iPad read them to you aloud. Enjoy full-color photo galleries and embedded video content, and access the Gazette's archives back to 2002. For only $29.99 a month, it's a great deal. Call 870-648-1209. That's 870-648-1209 to subscribe to the Texarkana Gazette today. Virologist Benjamin Neuman didn't intend to become one of the most trusted science communicators of the COVID-19 pandemic. But as the virus he helped name began to spread last year, requests from journalists and questions from ordinary people prompted him to begin sharing his expertise. Now his video explanations on Facebook and YouTube have stacked up thousands of subscribers and views. Dr. Neuman recently ended his stint as chair of the biology department at Texas A&M University, Texarkana, and began a professorship at the school's main campus in College Station. We spoke by phone about the science behind the virus and its vaccines, and what concerns him most at this stage of the pandemic. Here's my conversation with Dr. Ben Neuman. Good morning. Thanks for taking a little time to talk to us today.
Hey, happy to be here and happy to help. What can I do for you?
Well, it seems like the pandemic has been worsening over the past month or so. Just this morning, as we're recording this, we got the sad news that a Texas side police lieutenant died overnight from COVID. How worried are you about the state of the pandemic right now?
Yeah, I think when we normally look at the pandemic, we see numbers that go up or down, but like you say, it's important to remember that there's a person, often a really sick person, behind each of these numbers. Right now, the numbers are going up very dramatically. They actually haven't showed any sign of slowing down in Texas or in the U.S. in general. So hopefully at some point we'll get to some kind of peak and then some kind of resolution. But right now, I don't know that anybody is taking the steps that we would need to get there particularly. I mean, individuals are doing things, but as a population, we are very, very vulnerable, and I think it shows in what's happening.
What are the questions you're hearing from people right now? What are people out there worried about?
Yeah, so I don't know. I think if you go back a week, it was, what's the Delta variant? You know, what's this thing going to do to me? Is this the big bad wolf or, I don't know, one of the little pigs? How worried do I need to be? And the Delta variant is just the latest in the kind of assembly line of slightly mutated versions of SARS-CoV-2. It's not particularly different to the other versions that are out there, but it does seem to be a little bit better at spreading. So this is one that is spreading, and I was just looking into some numbers the other day about this. So I looked at the flu season numbers, and they're really amazing. Normally, the flu season has a great big peak. It's like a mountain where there's all kinds of cases, and they're right around the winter time, exactly where you'd expect. This past year, 2021, there is no peak there. It is a flat line with a few people getting it here and there, but absolutely no flu season. So flu is pretty much no longer an issue because of the steps we've taken to get rid of COVID, which is kind of a nice side benefit. And the other one is measles. So if you go back, let's see, it would be 2 years ago, we were having a rising number of cases of measles. And this was kind of as a result of the MMR vaccine and all the bad press that it was getting at the time, which was totally unfounded. But, you know, bad news travels faster than good news, I guess. And so there were over 1,000 cases in 2019. Get to 2020, and there were only 13 cases. And you get to 2021, and so far in the year there have only been 2 cases, at least as of yesterday, unless a new one has come in. And so that's another virus, and that's kind of true for all the other viruses that people get. The protocols that we've taken to knock out COVID and just being a little more careful about masking and distancing has pretty much wiped out all the other viruses. At a time when this is happening, and when every other virus is on the real decline, you see things like the Delta variant absolutely spiking. And so I think this tells you the story in a much more visual way. This is why we worry about the Delta variant, because even the protocols that we're following that take care of everything else are really not doing the job against Delta. It's a matter of vaccination, or we're just dealing with this terrible thing that's going to keep on changing.
What is it about the Delta variant that varies? What's different about the virus itself?
About 30 things.
Okay.
And of those, we have some understanding of, I would say, 3 of them, and really no understanding of the rest. So the discussion tends to focus on the things where it kind of makes sense.
Right.
So this is a virus that has one of the early changes. It's a thing called D614G. So this is one of the first changes that happened. It started out over in Europe. That's where they first picked it up. And it spread around the world. And within about 3 months of appearing, this was virtually 100% of the viruses that you could see out there. So it completely took over and outcompeted all the other viruses. this is weird to think about, but the viruses are kind of fighting over our lungs right now.
Right.
Yeah. And so it's built on that one, but so is everything else that's out there right now. And so we've learned that this makes the spike a little more stable. And it looks as though once the spike was stabilized, because the original version of the virus that came out was not very well adapted to humans, and not very stable, and just not very good at anything. And the virus, gives something time enough to learn, and nature finds a way, right?
Right.
It's like Jurassic Park. And this virus is doing that. It's just the totally random process of evolution. But the way a virus works, it doesn't just try one thing like a person would. We'd say, well, maybe I'll change it here. The virus is going to randomly make every possible change. And it does it at a rate where it's actually, once you get the virus inside you, within the first day, it will have made every single possible change, and it will have made every possible combination of 2 changes, and most every possible combination of 3 changes that could exist.
So we're talking about millions and millions of permutations.
Right, inside every infected person. And so nobody really gets one virus, or rather you get one. You start off with one, but then all of a sudden you're facing all the variants inside you, kind of. And the variant that you get is just kind of like the starting place for this little cloud of viruses that's going to develop. And so you get all this trivial variation in the virus. It's changing in every possible way. Usually these are bad for the virus. You can pull these out and show that 99% of them are dead, something like that. They just are broken fundamentally. But you'll get a few that can have some kind of extra power, like they're different enough to where they may not work as well, but to where an antibody that you made against the original version is now no longer going to be able to get as good a grip on them. And so this sort of interaction between the immune system and this sort of cloud of just spamming random changes is how the virus actually moves along. And so we end up with changes all over the genome. And so that's why I said there's about 3 we understand and, 27-ish that are there.
Right.
And eventually we could figure them out, I suppose. But, it's easier to just control this thing. I guess there isn't anything really different that I would do to stop the Delta variant. It still responds very well to the vaccines we have, and masks keep Delta out just about as well as anything else. It's the same size particle, so an efficient filtration system is going to work just as efficiently.
Tell me if I'm on the wrong track, but this is something I'm curious about. Wouldn't continued mutation result in a less deadly virus? I mean, it seems like quickly killing its host would select against a virus reproducing a lot. Am I on the right track there?
You're on the right track there, but the time scale is what is probably not right.
Okay.
So while we would be selecting the virus for one that can, not kill us, the virus is also going to be selecting us. And so you're going to get a change in the human population. And it takes us about 20, 25 years to get to childbearing age. So humans change very slowly, and the virus changes very quickly. And so the virus is always going to move faster. And so I think over the course, so to get to a situation like you're talking about, where the virus doesn't kill us and we don't get too sick, you have to change both the human population and the virus population.
I see. Okay.
And you're talking about something like 10,000 years would be a reasonable estimate for something like that. That's about how long we've been living with flu, we think. And flu is still rotten, and it still kills some people for sure. It's just not quite as bad as this.
Let's talk about vaccination a little bit. Another thing I'm curious about, as a scientific researcher yourself, do you ever think about those who created and developed the vaccines? Do you think they deserve to be better known and more celebrated?
I know at least of some of these people, and I know some of the people that did some of the testing on the vaccine and the early antivirals. Some of these people actually come to the coronavirus meetings, and they're just regular scientists. I don't know any of them that is really a super duper people person who would want to be, that statue in the park holding up a test tube.
They're not looking for fame.
No, and that's fine. Yeah, leave that to Hollywood people and politicians, I guess. But I think, I think they deserve a lot of credit. When it first started out, those mRNA vaccines in particular did not. They had been a great idea that had never quite panned out. And it was actually a couple other changes that were made by somebody who was trying to solve the structure of a spike. So the problem with these spikes that are on the outside of the virus is that they fall apart if you look at them wrong. I mean, you try to purify them. The best they're going to do is get about 10% of them that are somewhat intact. And even those, if the pH goes a little up or down, or you get your salt concentration just 5% wrong, they're all just going to burst.
We're talking about proteins, correct?
We're talking about a protein, which is just a big long chain of amino acids. That's what all the machines in biology are made out of, basically. Yeah.
Okay.
Yeah. But this is a protein. It's like, I don't know, there are flowers called jewelweed or touch-me-not. And when you touch the seed pod, it just explodes because it's like a little coiled spring in there.
Okay.
The spike is kind of like one of those, or like one of those party poppers, if you like, that you use on New Year's Eve.
Right.
It's got a ton of stored energy inside of it because the way the spike works is it's actually going to go through several different shape changes. It's going to transform and basically turn itself inside out, hook into the cell the virus is trying to enter, and then reel the virus into the cell and sort of crash it with a lot of force, all while coordinating with a bunch of other spikes to do this. So it's a coiled spring of a machine, and it is really hard to even purify virus and get the spikes still attached to it. But they figured out that there's a little place in there where you can make a few changes. So you can change some of the activation sites. And they did that. And then you can make a couple other changes that are just in one of the spots that needs to be able to move for the spike to unfold. And if you make the right change there, it turns out you get a spike that's something like 10 times more stable, and you can make 50 times as much of it, something like that. And so the way the vaccines worked is they took these changes, which were made for the MERS coronavirus, totally different coronavirus spike, mapped them onto the SARS coronavirus spike, figured out where they need to be, made them, tried it, and you know what? It worked. It just happened to work really well. And that's probably the key that made RNA vaccines in general and these specific vaccines work. Everybody is using this same version of what they call a stabilized spike. Every single vaccine that's out there.
What about vaccine hesitancy? Are there legitimate questions out there? There tends to be this sort of stereotype that people who are hesitant to get vaccinated are cranks or conspiracy theorists. What questions are you hearing from people that really deserve a compassionate answer, I guess is the question.
Yeah. I mean, that's probably the only way to really connect with anybody, right? Mm-hmm. Yeah, just telling somebody you're wrong. Nobody wants to hear that about anything. This is the whole reason why police have a hard job. That's basically what they do.
Right.
And so, yeah, I think that's probably the only approach. But at the same time, I don't know that anything that I would say would necessarily connect with somebody who is vaccine hesitant, because the way it seems to work is that you start out by mistrusting everybody on the inside, if you like, which ends up being everybody who would actually know what they're talking about. Mm-hmm. And you move from there, and once you've ruled out, sensible information, there's a world of nonsense out there, but,
right.
It's kind of an unlimited world, and that's all there is to pick from. And it's hard to get through. I think where there has been success, it's been with somebody that's already in that group talking to people.
Right.
Like there was a very nice podcast, I don't even remember which one it was, I think maybe a Radio Lab, where they had Chris Christie, the former governor of New Jersey.
Mm-hmm. I think I heard this too.
Yeah, exactly. And talking to people who were registered Republican, had voted for President Trump, and were at least vaccine hesitant, but said they weren't maybe completely against the vaccine. But boy, when you heard him talk, they were.
Yeah.
And hearing it from somebody that you already trust, I think, is probably the key. So there are a lot of things I hear out there, but these are not, these are all essentially fiction. They're sort of invented stories, and they're from people that are worried about something or that don't trust somebody. And I don't know that playing whack-a-mole with those things is necessarily productive, because it's really easy to imagine more. And kind of built into that, you'd say, well, why should I listen to you?
Right.
When I already believe this thing. Yeah. So, I understand it, but that's a really hard thing to push back against. And I don't know. The approach I've been taking is, if you find somebody that's willing to talk to them. And if they have questions, try to answer their questions, because I think there are doubts. There are things that people don't know. And in a lot of cases, this is stuff where somebody has done an experiment and has actually come up with a real, honest to goodness answer for it. The thing is, these are buried in science papers that are super numerous at the moment, and they can be a little dense, a little hard going. So I figure that, if you can get people who have to read these science papers and know what they're about anyway, and get them to answer questions, I feel like that's probably going to be the most effective thing. But, we haven't solved it yet, so maybe there's a better answer out there, and I'm listening.
What can you tell from the virus itself about this question of whether it was somehow engineered by people? Can you just look at the virus and determine anything about that?
Short answer, no.
Okay.
Longer answer, you can make a pretty good assumption.
Okay.
So when people are worried about controversies, lab leak, whatever, something like that, they are generally focusing their attention in a very, very narrow way. So they're usually looking at two parts of the spike. When the spike makes up, a little less than 10% of the whole virus genome, and the parts that they're talking about are maybe 5%, or actually a little bit less than that, probably about 3% of the genome. And so we're focusing on these very little things. And generally the conversation says, well, we looked at a regular SARS coronavirus, like the original one, and we looked at this new virus, and here's the difference. And you can say, yeah, for sure, there is a difference at these two spots. But if you zoom out a little bit, you find all of these same features. You find receptor binding domains that swap out and recombine in every single coronavirus that's out there. In the chicken coronaviruses, which, there are probably people who would listen to this who would be poultry farmers. So we're talking about infectious bronchitis or IB, sometimes I know the farmers will call it. But not IBDV. Yeah, that's a different one.
Okay.
And there are just so many of these out there, and they're always popping up. And, for that one, the vaccines are actually not great, and a lot of people don't vaccinate. But there's this just sort of rich coronavirus literature out there. And if you take the bigger view, there's nothing on the new virus that we haven't seen over and over again on hundreds of other coronaviruses. These are just solutions that nature comes up with constantly. And I think the other way to look at it is that we tend to look at a few mutations in the spike and say, oh, these are the evidence because I see them here, I don't see them there, and so therefore somebody put them from here into there. Yeah. But the nearest virus that we know is about 1,000 nucleotides different. And so several hundred of those, maybe 950 or so, are found outside the spike gene. And these are found in the usual places. So there are some genes that never change very much from one coronavirus to another, and those are virtually intact with just a few changes. There are other genes that change colossally between one virus and another, and in those you find a lot of these changes. And the changes look like just random changes. They're not things we've ever seen before. And so the only way you can get from a virus that you know, like the ones that were sequenced a couple of years ago that are kind of similar but 1,000 nucleotides different, the only way to get from that to a virus like SARS-CoV-2 is with a whole lot of evolution, just regular mutation, selection. Most of the mutations are going to die out, and we know that this virus changes at a rate of about 30 mutations a year, and it's been doing exactly that throughout the pandemic. And so you say, all right, how many years does it take to make 1,000 mutations? And then you say, okay, how long have we even known about coronaviruses? Right. And you come up with these answers where, wow, there's really no way. And there's no way to speed up this process that anybody has really found. So it's just not a thing that could happen. I would say some people tend to look at those 3 or 4 mutations. I tend to look at the other 900 and some mutations, and they look like just one more of the many naturally occurring coronaviruses out there. And they don't look like anything that was at the Wuhan Institute of Virology. So based on that, I've got to say it looks like something that came from nature.
So these viruses, coronaviruses generally, and other viruses are out there in wildlife populations going through this process of natural selection. How worried are you about that zoonotic transmission from animals to humans? Is this something we're going to see a lot more of? How can it be prevented?
Right. This is a thing we've seen happen 2 more times I think it was early 2021. So there was something that was kind of halfway between the dog coronavirus and the cat coronavirus, but it picked up just enough mutations that it actually made a person sick. And if a virus can get into a person and make them sick, then there's a pretty good chance that, there's nothing that special about any one of us, unfortunately.
Right.
So it probably has the potential to spread. There are a lot of cat and dog coronaviruses out there. We usually don't vaccinate for them. We actually don't even have great vaccines for those things. And so that's a risk because, we have pets. I have pets. I have cats and dogs. Yeah. Mm-hmm. So, the potential is there.
And not to mention farming.
Yeah. Oh, my goodness, yeah. So there are cow coronaviruses. Those are closer to some of the mild human common cold viruses. And those also appear to go back, if you go back far enough, to bat viruses. Yeah, you got your pig coronaviruses. There was a porcine Delta coronavirus that's never been seen in humans before, and all of a sudden it jumps and makes several people sick this past year. So this is a thing. We live in a world of near misses. And thank goodness our immune systems work really well. But think back to all the times when you've had a sniffle or a cold. How many times did you go somewhere and get a molecular test that said exactly which virus you had?
Right.
And a molecular test that would have picked up new viruses that we didn't know about. There are ways to do this. There are parts of sort of generic classes of virus that you can look for. But those are expensive tests, and this is not what you're going to get at your, friendly neighborhood doctor, and because it's not what we normally need. But yeah, some percentage of those are probably exotic things that, for one reason or another, and thank goodness, just never really took hold in humans and never really spread well.
Should we be taking steps to limit contact with animals? What's to be done about this?
Right, but every kind of animal that we have looked in has its own coronaviruses. We have coronaviruses now in lots of ocean fish and in frogs and in salamanders and in virtually any mammal or bird that you would look at.
So there are umpteen, would you call them species or varieties that have been identified, and that's just the tip of the iceberg, correct?
Right. We've only seen the ones we've really looked for, and there are a lot more places still to look. So, there's a lot of different viruses out there. And it's not just the coronavirus world either. There are all these other groups of viruses. Ones that are related to polio virus are actually far more common. And, it seems like pretty much everything out there has one of these at some point in its life, anywhere that you'd look. Yeah.
So I guess the action plan, so to speak, shifts to how to respond when inevitably this transmission happens. Yeah.
I don't think there's any realistic way to prevent the first case, but you can shut this thing down at 1 or 2 or a few cases. Okay. I think we took a very laissez-faire approach at the beginning. And you saw the U.S. trying to invent its own test for, I think, political reasons, because we didn't want to use the test that some German colleagues of mine actually came up with, which was fine. We ended up coming with pretty much the same thing, but it put a delay on there when we couldn't really test. And we tried targeted banning on people from certain countries when you know full well that planes, bounce around between all kinds of countries. And yeah, you either got to completely stop travel, or better yet, you just do more testing when there's one of these things around. And even before it gets to that stage, when you've got a person sick and you don't know what it is, there are steps that can be taken now, tests that can be done to figure out which kind of virus it is. And I'd like to see those get out to more places, so that more doctors and hospitals actually have access to this. I think that may be the big step. Then that and good contact tracing, which we're still not very good at, can really shut down anything.
So similar to cancer, early detection is very important.
For sure. Yeah, viruses go viral. Yeah. And you want to catch them before they do. Yeah.
So did you ever see yourself becoming such a popular science communicator?
No.
How did that happen?
Yeah. I was a little nervous about it at first. I had done a couple of things. I think San Diego was the first time I did sort of a virus news story, and I was just frightened. But it was SARS, and we were the lab that was working with it, like original SARS, back in 03, 04. And so they just wanted some kind of picture for the news, and so my boss didn't want to do it. He said, "Hey, you're gonna do this. Just show them your slides that you made, your PowerPoint with the pictures of some of the parts and how they work, and talk about it." And I think they ended up doing a voiceover over most of the stuff that I said.
Right.
You know what? It was fine. I really got more into it in England when I was working there at the University of Reading. They had a really good press office. It was a bunch of guys that had just gotten fired by basically the newspaper industry as it was really starting to contract. And so they had all these really good high-end reporters, people that knew news, and they ran this thing like the White House press room.
Oh, wow.
They had 10 TVs going on. So, actually it was more like 5 because there's like so many channels over there that show anything. And so if anything was in the news, they would get on the phone to somebody at the university and say, "I know this isn't quite your area, but it's kind of, can we plug you in?" And then they would call the producer and say, "We've got somebody who could maybe fill time." And I think what most of these producers were looking for in most cases was just somebody who could explain something. And I don't know, if you're any good at teaching, that should be what you can do.
Right.
And so, yeah, it was just a matter of getting plugged in. And then it was really helping out the university. It ended up helping me out as well. Yeah, it's, I don't know, free advertising in one sense for other people, but I feel like there are better reasons to do it than for free advertising.
Yeah.
Just because otherwise it's hard. Imagine you're a news anchor and you've got to cover wars in Africa. You've got to cover, what's going on with the stock market. You got to cover the latest developments at the UN, and then you've got to switch over and put your molecular biology hat on and, cover some new virus.
Oh, believe me, I understand that.
I don't think there's anybody that has that many hats. Yeah.
Mm-hmm.
That's an Imelda Marcos load of hats.
Right.
And, so it helps to get in experts that can at least get close. Try to sort of make sure that whatever's coming out is not just sort of guesses and rumors and nonsense.
So I'm curious about any insights you've gained over the past year and a half. How would you rate American scientific literacy generally? I'm sure this is something you encounter in teaching too.
Yeah. I mean, the high end is really high, and it's weird that I feel like I have to defend science when when all science is the stuff that we've tried enough to know whether it works or not. If you're testing to see whether a light bulb is blown, and you switch out a light bulb from another lamp that you know works and find that it doesn't work. You've done an experiment. That's basically science.
Mm-hmm.
And there is a weird kind of anti-science backlash. The first time I noticed it was really with the climate change stuff. People were saying, well, that's just a theory, and you don't know what you're talking about, that sort of thing.
Mm-hmm.
And it probably goes, I mean, you can find this back in the 1800s and the 1700s, anti-vaxxers. As soon as we had a vaccine, there were people that were opposed to the vaccine.
Right.
So, yeah, Jenner was dealing with these things as we are today. And so I don't know if it ever went away, but maybe that's the first time that I've noticed it. And it's it seems to be the same people that would say that climate change isn't happening, but are also saying viruses aren't happening. And I don't know, it seems like wishful thinking to me.
Mm-hmm.
But it also seems like a position that's taken for a purpose. I don't know. I don't think anybody necessarily starts out believing any of that. You just sort of grow into it over time, would be my guess. I know people who don't have the vaccine, who don't particularly think the virus is real. I'm related to some people like this.
Mm-hmm.
I need to spend more time and try and get inside their heads, and I can give you a better answer.
Well, you can't be a psychologist, too.
Yeah, or rather, I could train and do that, but at the present, I would not be a good one.
So you've left us here in Texarkana. Tell us about your new position. Are you living in College Station proper now?
I am, yes.
Okay. What are you going to be doing at A&M?
We are actually opening up some of the samples today. We're going to be growing some live virus, and we're testing it out on a few different cell types to see which one grows the virus best. So we're going to be doing some antiviral testing, but step one of antiviral testing is you've got to have a virus that grows a lot.
Mm-hmm.
Because if you can get 1 million viruses per mil out, then you can knock it down, 1 million-fold if your drug is really awesome.
Right.
So just gives you more room to play with and maybe find something that works better.
So this could have real practical applications down the road.
We are very much hoping, but these are early steps, and most of the early steps end up leading nowhere, and you just got to make a whole bunch of them to have a reasonable chance that something pans out. That is science, essentially.
And that's life.
There you go, yeah.
Thanks so much for taking a little time to talk with us today. That's been very interesting, and I think people are going to learn a lot from it.
Well, I hope so.
We'll talk to you again later down the road, I hope.
Happy to do that. Thanks very much.
Okay, thank you very much.
Yep, always happy to help.
On the Line is a Texarkana Gazette podcast recorded in our newsroom Star Bear Studio, right here in downtown Texarkana, USA. The show was written, produced, and edited by Carl Richter. News stories are based on Gazette staff reports. Texarkana Gazette podcasts are made possible in part by a grant from the Google News Initiative Journalism Emergency Relief Fund. I'll see you next week on The Line.